Okay, so have you ever stopped to wonder how our bodies pull off the same exact job in totally different places? Like in the brain versus the liver? Well, today we're diving into biology's incredibly smart solution for this, a special group of proteins called isozymes. And trust me, they are the true masters of adaptation.
So let's kick things off with a really central question. Inside the amazing, complex machine that is our cells, can one specific biochemical job actually be done by more than one enzyme? Is there some kind of backup system, or maybe a specialist for every single situation?
Well, the answer is a huge, resounding yes. And it's actually fundamental to how all complex life works. Biology is just packed with these specialist molecules, and they give our bodies this incredible flexibility. And of course, they have a name.
They are called isozymes. Sometimes you'll hear them called isoenzymes, but it's the same thing. And the definition itself is pretty simple, but wow, is it powerful. They are just different versions of an enzyme that do the exact same chemical reaction, but, and this is the key, they have different physical structures. And it's that difference in structure that unlocks all of their unique abilities.
Here's the way to think about it. Imagine you need to drive a nail into a wall. You could use a massive sledgehammer, or you could use a tiny little tack hammer. Right? They both do the same basic job, but you would never use them for the same project. Well, isozymes are basically the cell's different hammers, each one perfectly designed for a very specific task.
So here's where it gets really interesting. On one hand, isoenzymes can do the same reaction because let's call it the business end of the enzyme, the active site, is pretty much the same. But on the other hand, the rest of the molecule is built from a slightly different recipe of amino acids. This changes their overall shape, how efficient they are, and, crucially, how they're switched on and off inside the cell.
Let's see what those different kinetic properties actually look like in the real world. So take these two made-up isozymes. Isozyme A, let's say it's in the brain, has a low Km. All that means is it has a really high attraction to its target molecule. It's sticky, so it can work super effectively even when there's not much of that molecule around. Now, isozyme B, over in the liver, has a high Vmax. That means its top speed is incredibly fast. It's an absolute workhorse, built to process huge amounts of stuff very, very quickly. See? Same job, completely different performance specs.
So you might be wondering, where did this brilliant system even come from? Well, it turns out it's a product of one of evolution's most powerful tricks.
It pretty much all comes down to something called gene duplication. Just think of it like an accidental copy and paste mistake when our DNA is replicating. All of a sudden, you've got the original essential gene, and you've got this extra spare copy. Now, this spare copy is totally free to collect mutations over millions of years without messing up the original's important job. And eventually, after enough changes, you get a brand new gene that codes for an isozyme, a specialist with cool, new, and useful properties.
Okay, theory is great, but let's make this concrete with a real-world example. The absolute classic, the one you see in every textbook, is an enzyme called lactate dehydrogenase, or just LDH. LDH is absolutely critical for producing energy in our bodies. Now, the enzyme itself is a tetramer, which is just a fancy way of saying it's built from 4 smaller protein parts, or subunits, all stuck together. And here's where the genius comes in. There are 2 different kinds of subunits it can be built from, an H-type, found mostly in the heart, and an M-type, found in muscle.
And this just illustrates the concept perfectly. By mixing and matching these 2 H&M building blocks in different combinations, our body creates 5 distinct LDH isozymes. LDH1, which is made of 4 H subunits, is perfectly tailored for the oxygen-rich environment of the heart. On the flip side, LDH5, made of 4 M subunits, is designed for the low oxygen conditions you find in hard-working skeletal muscle. And the other 3? They're just hybrids, each one optimized for the specific tissue where it lives. It's brilliant.
Just take a second to think about that. From only 2 basic building blocks, nature creates 5 completely unique, specialized enzymes. It's an incredibly elegant and efficient system, right? Creating all this functional diversity from a very, very limited set of genetic parts.
Now, because these isozymes have different mixes of H and M parts, they all have slightly different overall electrical charges. And this is super useful because it allows scientists to separate them using a lab technique called electrophoresis, which basically just pulls the charged molecules through a gel at different speeds. And this isn't just some neat academic exercise. It's a critical diagnostic tool. When someone has a heart attack, their damaged heart cells release LDH1 into the blood. So by measuring the levels of that specific isozyme, doctors can actually confirm that there's been damage to the heart muscle.
Okay, so let's pull all of this together. Why does the body even go to all this trouble? What's the real payoff of having isozymes in the first place?
At the end of the day, isozymes are a fundamental strategy for control and adaptation. It's how different tissues get enzymes perfectly tuned to their unique jobs. It gives our whole metabolism flexibility. They can even be switched out at different stages of our development. And, like we just saw with LDH, they can be powerful signals that something is wrong, acting as diagnostic markers for disease.
If you take just one thing away today, let it be this simple little formula. Isozymes mean you get the same reaction, but with a different structure, and that allows for highly specialized regulation. It's how biology achieves both consistency and incredible flexibility all at the same time.
And that kind of leaves us with one final thought. This whole model, using slightly different versions of the same basic part to build complexity, it's incredibly powerful. So where else in biology do you think this is happening? You know, from our immune system to the neurons in our brain, where else might nature be using this same specialist strategy to fine-tune its most critical functions? It's a pretty fascinating question. Thanks so much for joining us.